Hi everyone. So this is today's topic is cleavage, and welcome back to my channel again. So cleavage is the part of developmental biology. Okay. So after fertilization, what happens? The zygote. After fertilization, the zygote that continuously divides. Cell division is occurred continuously. Okay. So this is called the cleavage. Cleavage actually the multicellular organism. How the from one cell the multicellular organism is produced is the process known as cleavage. Okay, so here we learn that the cleavage cells are called the blastomeres. Okay, cleavage cells are known as the blastomeres. From egg cytoplasm to numerous smaller nucleated cells arise. Okay, the process by which cleavage. Clear? So cleavage pattern, it is dependent in two parameters. Okay, one parameter is the distribution and the amount of yolk protein in cytoplasm, and the second one the factors of the egg cytoplasm that influence the angle of the mitotic spindle and the timing of its formation. So two parameters are what? One parameter is the amount and the distribution of the yolk, and the second one is the some factors that present within the cytoplasm that determine the timing and the procedure of cleavage. Clear? So like that, there is a egg cell. There is an egg cell, and here we know the two part in that cell. Two part. One is the vegetal pole and another is the animal pole. Why it is vegetal pole? Vegetal pole because there is the amount of yolk is maximum. So this is called the vegetal pole, and here the nucleus is present, and this is called the animal pole. Clear? And now we learn that is the which one horizontal plane and which one is the lateral plane, and this is the vertical plane. Clear? So here this line is showing the horizontal plane, how the cleavage pattern is occurring here. We just learn in next part. Okay, so here we just learn about the vegetal pole, animal pole, and the planes. Okay, this is the horizontal plane. In such a way, when the cleavage pattern are arised, so this is known as the horizontal plane. This plane is known as the lateral plane, and this is known as the vertical. But vertical, the yolk phase, from yolk phase, this is ventral. Okay, ventral form, and this is the dorsal form. Dorsal form. Clear? So this is the actually structure of an egg, and how the cleavage patterns are arised we learn here. Okay, so this is actually cleavage, and cleavage is very important because without cleavage the mitotic division is not possible, and from zygote the one cell that cannot transfer into the multicellular part. Okay, so this is the process by which egg cytoplasm that changes to the numerous smaller nucleated cells, and the pattern is totally dependent on the yolk material and the some factors that present in the egg cytoplasm. Okay, now we learn about what types of cleavage. That means what pattern of cleavage we can see in different type of animals or the species. Clear? Okay. Now this is different types of cleavage pattern we see here. First one is the holoblastic. In case of holoblastic, the cleavage is complete here. Okay, holoblastic. In case of holoblastic, cleavage is complete here. And under holoblastic, these are the types. One is isolacital, and second one is mesolacital, and the other parts are also there, but there is a space is very short, so that's why first we learn about this, and then another part we learn. Okay, so this is isolacital. In case of isolacital, the distribution of yolk is evenly. That means the total area is evenly distributed with yolk. Okay, so that's why it is known as the isolecical. Now here the isolecical, under the isolecical we see here the different types of cleavage pattern. One is radial, second one is the spiral, third one is the bilateral, fourth one is the rotational. Okay, and in case of radial we see this kind of cleavage pattern. Okay, this kind of cleavage pattern we can see here, and the example is echinoderm. The second one is the spiral. Here you can see that the cleavage pattern is like this. Okay, cleavage pattern is like this, and this we can see this pattern in annelids. Third one is the bilateral, and this present in the tunicates, and this kind of cleavage pattern we can see there. Fourth one is the rotational, and it is this pattern is present in mammals, and the pattern is like this. Okay. So these are all the types of cleavage pattern that is present in the isolecical. Okay, isolecical. The second one is the mesolacital. What is the meaning of the mesolacital? The moderate yolk deposition. The yolk deposition is moderate. And under this one kind of cleavage pattern we can see, this is the displaced radial cleavage pattern, and the example is amphibian. Okay, example is amphibian, and here the cleavage pattern like this. Okay, so these are the types of cleavage pattern that we see in the holoblastic or the complete cleavage, and under holoblastic in the isolecical and the mesolacical. These are the examples and these are the cleavage pattern. Now we learn another parts. Okay. Okay, now we learn about the Neuroblastic cleavage. What is Neuroblastic cleavage? First one is the holoblastic cleavage, where the cleavage was complete, and this in case of Neuroblastic cleavage, the cleavage is incomplete. Okay, under this we can see this pattern of cleavage. First we learn about the Neuroblastic. Under Neuroblastic, what type of cleavage we can see? One is the telolacical, and the second one is the centrolacical. Centrolacical means actually in case of telolacical, the dense yolk throughout the cell, but telo means actually the tail part of the egg that is totally condensed with the yolk material. Okay, and in case of centrolacical, the central portion, okay, central portion is occupied by the yolk material. Clear? Under this telolacical, we see two types of cleavage pattern. One is the bilateral, and second one is the discoidal. In case of bilateral, the cleavage pattern is like this. It is superficial, and the cleavage pattern only occurs in the central region. Okay, in the central region, and it actually superficial, or that means the cleavage pattern not entirely cell, it just cannot enter the depth of the cell. Okay, so that's why it is superficial. And in case of discoidal, an example is cephalopod. And in case of discoidal, the cleavage pattern is like this, disc shaped. Okay, disc shaped. You can see that disc shaped. And example is fish and bird. And the centrolacical cleavage pattern generally also superficial, and it is seen in insects. Okay, and this type of pattern we can see here. So these are the total pattern we learned here, the cleavage pattern, and what kind of animals bear this type of cleavage pattern we learned here. Okay, in the next video we learn about the definitions or the explanation of the different cleavage patterns. Okay, thank you. Okay, now we explain about the radial, spiral and bilateral rotational cleavage. Okay, so this is the second part of the cleavage pattern. Okay, and you just here before that I explained about the different type of pattern and how they are situated. So this is the second part, that is the explanation of different types of cleavage pattern. the first one is the radial cleavage, and here the in case of radial cleavage, the embryo is divided right angles to the previous one. Okay, embryo actually divided right angles to the previous one. This is a radial cleavage, and like that. As the first one is here, like the division is now, the second one division is that. Okay, so this is the radial cleavage. The second one is the spiral cleavage, and here the arrangement of the blastomeres of each upper tier over the cell junction of the next lower tier, and the spiral around is seen. Actually, here the blastomeres, the first blastomeres that are produced in first situation, they are situated in the upper tier and over the cell junction of the next lower tier. That means here, after formation of the blastomeres, one blastomeres remain in the lower tier and the another blastomeres remain in the upper tier, like that. Okay, so this is situated like this. That means here the blastomeres arranged in such a way that it looks like a spiral pattern. Okay, spiral pattern. And the third one is the bilateral, and here this is a complete division, and after the first cleavage, dissection of the zygote into left and right. So here when the first cleavage is occurred, then you can see that the two blastomeres or two daughter cells divided in such a way. One is present in the left side and that is present in the right side. The fourth one is the rotational, and here a normal first division along the meridional axis. Meridional axis means as it is animal pole, as it is vegetal pole, this axis is known as the meridional axis. So it involves a normal first division along the meridional axis, giving rise the two daughter cells, and then divide. That means this daughter cell that divide again meridionally. Clear? So here we can see the pattern is rotational. Clear? Next step is the mesolacital, and under mesolacital there is we learn the displaced radial, and in case of displaced radial, the first division begins at the animal pole. As it is animal pole and it is the vegetal pole, the division starts from the animal pole and it extends slowly down to the vegetal pole. As it is animal pole and as it is vegetal pole, so this starts from the animal pole and enters, slowly extends to the vegetal pole. So this is the displaced radial pattern. Another pattern we learn after that. Okay, under Neuroblastic Cleavage, there are two kinds of centrolacital and ankylolacital. The centrolacital, under centrolacital, the superficial cleavage is seen, and in that superficial cleavage, the divisions occur only in the rim of the cytoplasm around the periphery. Around the periphery, the rim of the cytoplasm, this is the periphery. You can see this is outside is called the periphery. Okay, this is periphery. So around the periphery, rim of the cytoplasm, around the periphery, we can see the cleavage pattern. In case of B, the telolacital, there is two kinds of cleavage pattern we have seen. One is the bilateral, another is the discoidal. In case of bilateral, the divisions occur bilaterally, and in case of discoidal, the divisions occur only in the disc of the cytoplasm. That is very little part of the cytoplasm and form the disc-like structure. So this is called the discoidal. So these are total completion of the cleavage pattern, and here I explain about the cleavage pattern, and before in this video, before I explain the different pattern, we just showing we have how the patterns are formed in the cleavage of the blastomeres and how formed this explained. Okay. So this is the cleavage pattern, and this is the main part of after fertilization, the zygote, what happened to the zygote. So cleavage is seen, and this is the actual cleavage pattern. We have seen the different types of individuals. Clear? So now I will discuss about different topic in my next video, and okay, thank you.